I'm good. So we will start, let's say, in a couple of minutes. Uh,
Yeah.
in maybe in about 5 minutes, we will start. And on behalf of my institute, our university, I would really like to thank you, Dr. Benjamin, for sparing his time to speak in this webinar series, which we have been conducting for the last few months. And we are very fortunate that Dr. Benjamin is here to speak on foodborne parasite, trichinella, and more about the genetic variations. And before starting, I would like to a bit about tell you a bit about Dr. Benjamin. So Dr. Benjamin Rosenthal is a biologist and epidemiologist seeking to limit the impact of zoonotic parasites on animal and human health. He has been he studied history and biology at Oberlin College and infectious disease epidemiology and parasitology at the Uni, Harvard School of Public Health. His work span questions in molecular epidemiology, systematics, parasite genetics and genomics, and evolutionary biology. He has made contribution to the study and management of nematode parasites such as trichinella and NP complexan protists such as toxoplasma, Neospora, sarcocystis, and Amelia. And his present work embraces a One Health perspective to establish poultry coccidiosis as a model to understand and prevent human cyclosporosis. Currently, Dr. Benjamin is associated with the Animal Parasitic Disease Lab, which is a part of United States Department of Agriculture. Uh, and now I would like to invite Dr. Benjamin to please speak on this topic. And once again, I welcome you, Dr. Benjamin, on behalf of our university as well as university officers and my department. Thank you very much.
Well, thank you very much, Rajneesh. And it's really a great pleasure to be able to, let's say, communicate with all of you. It's a fantastic opportunity, and really, there are quite a few of you. So it won't be possible for me to understand all of your individual motivations and interests, but I hope that some of what I have to say this evening for you, this morning for me, will be of some interest. And I thought that, understanding your overall orientation in this series to One Health, that I would think about one of the systems that my colleagues and I have been studying for some time that spans at least veterinary and human health. And of course, all zoonotic infections are important both from a standpoint of veterinary medicine and human medicine, public health. The environmental health portion comes into play as well. And I will just use this example to discuss some of the ways that a geneticist and an evolutionary biologist, a population geneticist, can contribute to understanding epidemiological processes, and we hope in helping derive solutions to epidemiological problems. So the title of today's talk, How Genetic Variation Helps Us Understand the Biology and Epidemiology of Foodborne Parasites, but I will focus my remarks today on trichinella. And before I do, I just want to acknowledge that much of the data that I'll be presenting and many of the ideas, the conclusions that I've drawn have come not from me alone, certainly, but through collaboration. So I'd just like to outline some very practical questions before we get into studies. Do animals infected with trichinella tend to harbor genetically diverse or genetically uniform cohorts of larvae? That seems like a simple question. If we have an animal and it's got a lot of parasites in it, are those parasites genetically the same or are they genetically different? I will say that we know from studying many different parasite systems that this answer is different from one parasite to the next. So another way of asking that question is how many pairs of parents were there and how related were those parents to one another in contributing to the larvae that you'll find in a given animal. And if you think about that further, it gets to an epidemiological and an ecological question. Did one or more meals likely account for the infection? And if you think about that, it relates to the immunology of the infection. If a given host, it could be a pig, it could be some other carnivore, is acquiring many different infections over the course of its lifetime, then it must not have a very protective immunity. But if an animal has what turns out to be a genetically uniform cohort, then that either means that infection is unlikely and the infected ones were those that happened to encounter an infection, or it means that they were protected from subsequent exposure. And then I'll just emphasize throughout this talk that getting answers to these questions from a given population sample may not tell you what's the case in other parts of the world. And I will also emphasize throughout that we know very, very little about what is the case in India. And I'm hoping through this talk to inspire somebody in the audience to make it his or her job to find out the answers to the questions where you are. There are very specific reasons for being interested in what's happening in India. So in thinking about these general kinds of questions about uniformity of infections, well, these bear on some practical things, like can we trace outbreaks of trichinella using genetic signatures? You know, tracing is not a novel concept. It's applied, widely in viral and bacterial epidemiology. It's used much less so in parasitology for some good reasons. But can we trace outbreaks? How much genetic information would we need? How much genetic variation would suffice in order to understand that a group of isolates were related to one another in a very specific way, or to exclude a suspected source as related to a given outbreak? And are these answers the same throughout the globe? So, just as a reminder, trichinella is a very unusual nematode parasite in that the only way one contract it is by consuming tissues of infected animals. The larval parasites insist in muscles, and they may remain for the life of that animal. It depends on the parasite and it depends on the host. Sometimes there are immune responses that kill these over time. In other cases, these are lifelong and they just remain. Nothing is excreted, nothing is urinated. There's no free living environmental stage. The parasite remains in the host. until predation or scavenging on the carcass of that animal induces larvae to mature to the adult stage, to pair up, to mate in the gut of the next host, to produce larvae that migrate through the lymphatics and the blood system and then establish new muscle parasites. So for human health, the greatest exposure risk is from transmission in pigs consumed as pork and developing muscle stages in the animal. So we've entered the age of trichinella genomics. I mean, the first draft genome of this parasite was published several years ago now. And that has led to a number of follow-on studies. And I will just mention a few of them, and some might work, some not. With my former postdoc, we were able to use a resequencing approach to identify previously unrecognized variation in mitochondrial genomes. I was just speaking with Dr. Sharma about his interest in sequencing mitochondrial genomes from trichinella and some Indian samples. So that continues. And work looking at next generation sequencing of related species of trichinella. This is the one that's found in wildlife in North America, and identifying greater variation in the wildlife parasite than in the parasite of domestic hosts. Work by Robin Gasser's group in Australia, sequencing mitochondrial genomes for all of the species in the genus. So this is organellar genomics. And then very importantly, draft nuclear genomes for every species of trichinella then known in 2016. Now, Dr. Sharma played an instrumental role in discovering a new species of trichinella, not included in this particular study because nobody knew about it, but has made a draft genome of the new one. So, okay, great. We now have genomes for these parasites. How can we go back to some of those basic questions that I outlined earlier? How can we apply genomic information to address some of those practical questions? Let's see. So I asked earlier, do animals infected with trichinella tend to harbor genetically diverse or genetically uniform cohorts of larvae? So if we were to take this animal and digest its tissues, much the way that stomachs digest tissues to break it down, we can look at the genetic diversity in that cohort of parasites. And work led by a Dutch group, Frits Fransen and Joke van de Giesen, with Italian and Polish colleagues, myself included, identified genetic evidence of interspecies introgression of mitochondrial genomes between Trichinella spiralis and Trichinella britovi. And here's just a figure of that. If you sequenced both nuclear genes and mitochondrial genes, what's being depicted here is a network of mitochondrial haplotypes. Two big circles there with some small little circles coming off. Those are different variants of mitochondria. But you can, what's, what is being illustrated by the long and actually broken dotted line is that there are two distinct versions of mitochondria consistent with these two different species. But there's a little bit of a problem. You see that some of the yellow is over on the blue side and some of the blue side is on the yellow side. Those colors denote the species that you would have diagnosed on the basis of nuclear variation in ribosomal genes. So there's been some swapping of mitochondrial genome among these two species. And if you were following Ernst Mayr's definition of species as independent reproductive units, then this should trouble you a little bit. How many parental pairs generally give rise to larval cohorts of Trichinella spiralis? Well, my colleagues in Italy developed means of genotyping individual larvae to investigate this population structure. So you imagine you digest some tissue and you identify lots of larval worms. And then you genotype each of the worms from that digest. Now, these are diploid individuals, just as you are. So at any given locus, they could have one or two alleles, just as you would be either homozygous or heterozygous at any given locus, depending on whether your mother and father gave you the same version of a gene or different versions of a gene. So I've just depicted those as two different colors. That worm is heterozygous at that locus. And then you can look at that locus in another worm and in other worms, and you can imagine that the maximum number of alleles you could conceivably identify is 2 times the number of worms that you genotype, assuming that this was a very diverse population. But this is actually much more typically what we found. When you characterize the same locus in this group of worms, they have 1 or maybe 2 different alleles. And this is a very busy table. I'll just point to you, what I basically just said. In EU1, which was 1 pig, characterizing 48 different larvae at several different loci, that's the columns, we found either absolute fixation, that is 1 allele at that locus, or in the case of, I think you can see my cursor, in this case, two alleles. So very little variation. And that was typically the case in pig after pig after pig. This was a locus that could vary, but it did vary somewhat over the entire population. But when you looked at the larvae from an individual pig, you found very few alleles. So just imagine now the parents giving rise to a collection of larvae. There's a mother and a father. How many alleles could one mating pair contribute? conceivably, she could contribute to, he could contribute to. So at most, we could imagine a given pair of parents contributing 4 alleles at a given locus. But how many alleles do we typically see in these larvae? 2 at most, or even 1. Now, what does it mean if we only see 1? Right? I mean, this wasn't always the case, and we have more sophisticated means of evaluating the number of mating pairs. But when it does occur, what does it mean that we find only 1 allele? Well, I think it means that there are many rounds of successive inbreeding, so that very closely related male and female parasites are mating and producing the next round of larvae. This is approaching the self-fertilization that some plants are capable of, where similarly you lose genetic variants with successive rounds of inbreeding. Now, if you'll indulge me some population genetics jargon for just a moment, considering 2 subpopulations, let's consider the extremes of genetic uniformity or difference. So down here on the right, we see that this population overall is composed of black and tan, beige, I don't know what that color is, black and yellow, let's call it, balls. But all the black balls are in one subpopulation and all the yellow balls are in another one. So now the reduction in total genetic variation attributable to the subpopulations is 100%. All of the genetic variation is explained by the fact that these 2 subpopulations exist. And geneticists call that an FST of 1 or 100%. In this theoretical example, we have the opposite situation where the alleles are distributed randomly with respect to the subpopulations. And now the fixation index is 0. So this value of FST ranges from 0 to 1. And I hope this gives you a sense of what that means. In statistical sense, it's the proportionate variation reduced as a consequence of the existence of these subdivisions. And there's a mathematical formulation that describes non-random occurrence of alleles within and between subpopulations, and they all get to 1. But so you can think about individual larvae that we're sampling and seeing where the alleles are, and consider an infected host to be the bounds of the subpopulation, and then the whole group of those hosts and their larvae is the total population. And I give you that background so that we have some intuition as to what it means when we look at the degree of inbreeding, considering cohorts of larvae, each cohort coming from its own host animal. And what I want you to see here, and I've just indicated some in red, is that when you compare the larvae of one host to the larvae in the other host, you can see sometimes that 70, 80% or more of the total variation is explained by the fact that larvae are sampled one host at a time. It's another way of saying that the one animal's larvae are very different from the other. I want you to also see that it's the case that you can encounter pairs of animals whose larvae are hardly different from one another at all. And this is true even in Europe, which has very little genetic variation at all in Trichinella spiralis. Still, the larvae in a given animal are so similar to one another that after characterizing a few of them, you can very well predict what the next ones will look like. In a sense, you could say, why did they waste all that time genotyping 48 larvae when they could have known from the first 5 what the rest of them were likely to be? Well, you wouldn't know that until performing this study, but that's the net consequence of the finding. And now I'm going to start talking about differences in global variation. This is a principal coordinate analysis showing the distribution of genetic variation. But here's something that I think is actually rather remarkable. In this group of isolates, dubbed Europe, North America, and South America, we are looking at genetic variation among larvae from many different pigs in, let's say, the West, in Europe and in the Americas. And they have some genetic variability, but they also have some genetic coherence. They are, it appears, one another's closest relatives. Here, what I'm showing you is the spread of genetic variation in larvae from a single pig in China. And these are from another pig in China, and another, and another, and another. And so you can see that the genetic variation among the larvae from one pig in China is comparable to or greater than the genetic variation from throughout Europe and the Americas. And now you're going to be seeing lots of figures labeled Asia, but I want you to understand that this is Southeast Asia. This is mostly China and some Thailand and some Korea. Nothing from the Indian subcontinent. Well, can genomics tell us more? Remembering that eukaryotes have a membrane-bound nucleus with most of their genetic inheritance, and remembering that we also have this organellar genome, remembering that your nuclear DNA is a composite contributed by both of your parents, by all 4 of your grandparents, by all 8 of your great-great-grandparents, but that your mitochondrial DNA came from your mother and came from her mother and her mother and her mother. So the mitochondrial genome is a non-recombinant, uniparentally inherited genome. Your nuclear genome samples over all of your ancestors. So I said the age of Trichinella genomics started with mitochondrial genomics, and here is just an alignment of a portion of the mitochondrial genome from a bunch of isolates from Europe and the United States. And these columns of color tell you immediately, it doesn't matter if it's an A or a G or a C or a T. The point is the uniformity here, that something from Poland or Finland or the United States or Argentina has the same mitochondrial genome at almost every position. There's some more variation in the D loop, but it's also harder to sequence. But here you see that here's a base that distinguishes some isolates from the rest. But you have to look far and wide to find them. Sometimes from an animal, we see that larvae include those that had more than one mother. But and more often in Asia than in the West. But then looking at groups of isolates that were separated by hundreds of miles in 20 years, finding only one difference in the entire mitochondrial genome. More variation in the mitochondrial genomes from East Asia. Not many, but more. We used information like this to construct a biogeographic hypothesis of Trichinella spiralis, essentially saying that all of those in Europe have a recent common ancestor and have expanded in an almost clonal fashion and disseminated to the dependence of Europe to the West. Whereas in regions closer to the origins of domesticated, the older origins and independent origins of domesticated pigs, in China, there's been a long-standing larger population of parasites. So where does that leave us? Mitochondrial genomes can help define an outbreak strain, but more than one haplotype can occur in the same outbreak. More than one haplotype can occur in an isolate. And in Europe and America, you could sequence the entire molecule and lack a basis for confident assignment. Prospects are better in East Asia. How about in India? I'd love to know. So what about the nuclear genome? This is a phylogeny reconstructed from almost 20 million bases of chromosome 1 of Trichinella spiralis. And I've kept this color scheme that I used a little earlier. The red ones are in East Asia. And those longer branches, again, tell you that there's much more genetic diversity in that group. It shows that they are a distinct group from those in Europe and the United States. The length of those branches indicates that one in East Asia is likely to be much more different from others in East Asia. Whereas picking any of the isolates from Europe or the United States will reveal very, very few differences from any others in that region. And here I've just added these bars that kind of indicate the relative genetic depth of each of those groups. And you could even argue, are the European and American isolates different groups or not. Maybe yes, maybe no. But just another way of illustrating that, nucleotide diversity in Chinese isolates was almost 40 times higher than that in European and American isolates. Much more heterozygosity in those diploid nuclear genomes. And then this is just a Venn diagram showing the overlap and the independence of variable nucleotide positions. So this is the beginnings of comparative genomics as applied to trichinella. And we can see that these are very different histories, very different levels of variation. Now let's go back to a more practical question. Understanding these differences in genomic variation, how easily can you trace outbreaks? Well, I would contend it depends on where you are. In our modest sample, I mean, which is to remind us all that what we think is going on depends on what we've sampled. More than 70,000 bases vary on chromosome 1 from at least one isolate to others. And only half of those variable positions occur in Europe. So is that a lot? Is that a little? I mean, 70,000 sounds like a lot. Assuming the future sampling bears this out, 70,000 bases, and so that's getting down to, well, in 20 million, that's fewer than 4 in 1,000 bases varying. So comparing chromosome 1 of one European isolate to the other, you might find 15,000 differences. And 15,000 sounds like a lot. I wouldn't have to sit here and count from 1 to 15,000. But 15,000 in millions and millions of bases is not so great, unless you have a very efficient system of genotyping. I mean, 15,000 in 20 million is about 80 bases per 100,000. And some isolates differ by even less than that, by about half. What about variation within an outbreak? I'm getting some back. I hope you can mute that. So within an outbreak, we found only 10 bases per 100,000 varying. And remember, that's true variation plus any sequencing error that we may have incorporated. So what is our power to use genomics to trace outbreaks? Well, it's easier in East Asia where parasites vary to a much greater extent. It's like if you have a very diverse room full of people, it's easier to pick out one from the other. Unrelated isolates in Europe may differ at several times as many positions as do isolates from a given outbreak. So that's a basis for discriminating among outbreaks. But the problem is that you need hundreds of thousands, millions of high quality base calls in order to see that. And that is achievable with new technology, but not yet in practical routine use. In fact, my laboratory right at this moment is demonstrating the utility of RAD-seq to garner hundreds of thousands to millions of reads in a very efficient manner and readily discriminate among outbreaks. We can do this, and we can show that we can do it in just several days and at a reasonable cost once you have the investment in a high-powered sequencer to do it. So we're opening new doors, but we know that this is not yet for practical application in a routine clinical setting. So studying genomic variation has begun to inform us as to the history and geography of Trichinella spiralis. It shows us that diversity in Asia is great and not much elsewhere. Again, I'm going to say East Asia. Infections tend to represent a homogeneous subset of a region's diversity. There's only occasionally evidence for more than two parents and more than one mother. So we have beginnings of understanding of the diversity within an outbreak tend to be quite modest. Mitochondrial uniformity can be absolute. It isn't always, but that's typically the case. Within an outbreak, nuclear genes may differ by, this is the same way of saying one in 10,000 bases on average. And regional diversity may exceed that by 8-fold, which seems like a lot until you try it. You need to do a lot of sequencing in order to begin to be confident in these differences. In the future, such data will need to be married to functional analyses in order to better understand which portions of the genome are being most strongly conserved and which portions diversified. For the purposes of this talk, I've treated all nucleotides as though they were of equivalent function or no function whatsoever. And we know that that's not the case. This will inform and be informed by our understanding of biological function, or at least it should be. And with that, I look forward to any questions you might have.
Thank you very much, Dr. Benjamin. And now participants, please, you can ask questions. And you can ask questions in chat box if you want.
I mean, I'd be curious about anyone who is interested in these sorts of questions in other organisms. If you have epidemiological or genetic kinds of questions that you are working with and not working in Trichinella, that would be a perfectly interesting conversation to have. Or anything that I said that didn't quite make sense to you or that you disagree with, I would love your perspective. So I see one question: what's the practical aspect of all this study? How can we apply this knowledge in the field? Well, exactly. I mean, I think I tried to provide a setting for what we've learned about genetic and genomic variation so that we can see how we can apply it to outbreak tracing. I said we actually have enough of an understanding now, and we have technologies so that, practically speaking, I can say we're working very closely with colleagues in Poland, for example. Poland continues to have much more problems with Trichinella in their swine herd than we do in the United States. And there they have a situation where the veterinary services are required by law to conduct an investigation when there's a positive diagnosis, and to impel responses to prevent outbreaks in the future. And so you can imagine farmers saying, well, I don't know where this problem came from. Um, but if you can show, you know what, we sampled from some rats on your property, and they're also infected with this parasite. And you know what? They're infected with the same genotype of the parasite as the ones that we found in your pigs. You need to put your feed into a secure container that the rats can't get into, and you need to take measures to reduce the burden of rodent pests on your property. Or, you know, a farmer can get into a fight with her neighbor. Well, I had a clean farm, but then I took some pigs from my neighbor, and then I had a problem. Must be my neighbor's fault. Well, the tracing ability allows to understand whether there's an epidemiological linkage between one uh, and the next. I mentioned too, from a practical standpoint, that these genetic answers shed some light on basic immunology, because an animal, let's say it's a wild animal, that's uh, always just eating what it can, um, may inhabit an area where it's very likely, even at a young age, to encounter multiple exposures. But if it's only ending up with a genetically uniform larval population, then it's very likely that immunity protects it from subsequent exposures. I mean, you could say, or the last one in has displaced the earlier ones. Well, we can do experiments to figure that out. But um, to the extent that you're interested in veterinary immunology or adopting a One Health approach, you're interested in understanding human immunology as well, then these genetic data, I think, bear on that immunological process as well. From a practical standpoint, what we want to do is limit exposure of animals and people to these debilitating and sometimes life-threatening parasites. Um, Genetics is not the principal answer to reducing exposure. Okay, proper animal husbandry, preventing the animals that people eat from eating contaminated feed, is the way that country after country has protected people from exposure. But genetics does, I think, have a role to play in shedding light on epidemiological processes, on immunological processes, and can help suggest further prevention methods. At least I hope so.
Thanks, man. Thanks, Ben. So that was a really a good question by Simran, and thanks, Ben, for answering that query. Yeah, for example, we do have an outbreak of trichinella back in 2010 or 12, and then there was recent which occurred recently a couple of years back in northern part of India in Uttarakhand. So there, it was a feast, and about 54 people they ate boar meat. They didn't cook it well, so they used actually roasted meat. They eat it, and out of those 54, I think three or four they got sick. They do have the periorbital edema surrounding the eyes, and myalgia, fever, and that kind of stuff. But doing such kind of studies would be helpful to trace those outbreaks if you really want to do that. So that's what I would like to add to your question, to your answer. One more thing. So if we talk about developing nations like India, so doing like whole genome sequencing and doing this mitochondrial whole means this sequencing, it's going to be a bit expensive. So would you suggest some techniques which would be more accessible, or do you think would be more not that expensive, which we can use to trace back such kind of outbreaks or relate epidemiologically at least. Uh,
So one thing I would suggest is we need to figure out whether Indian parasite populations are more like Polish ones or more like Chinese ones, um, because what I've tried to emphasize here and to my colleagues more generally is that um, the efficacy of a technique depends entirely on the level of genetic variation that a population harbors. If you needed to differentiate among certain kinds of organisms, you don't need very much genetic information at all because there's such a high level of variability that you go just a short ways and you identify all the information you need. It's only in a situation where there's such uniformity that you need to search and search and search. So, step one is just understanding where you are, because you could actually waste a lot of effort by doing too much genotyping if that's not what's required. If you do need, let's say, European levels of genotyping in order to discriminate, then I have some technological fixes. We are using a system called RADseq, and there are similar approaches that say, you know what, you don't need to sequence the entire genome all over again. Here, use some restriction endonucleases, and I know you're very good at that. Instead of applying those endonucleases to one PCR product, I want you to apply those endonucleases to the genomic DNA and now construct libraries that contain random fragments of that digest and sequence those. You could save yourself lots on the sequencing reagents. You can, in fact, multiplex these. So you can, in one sequencing run, characterize those bits, that library, from a dozen or 20 different sources. And using some bioinformatic wizardry, then separate the sequencing results from each of those libraries and then compare, and allow you in a very efficient way without doing all of the resequencing, just maybe a tenth of it, maybe even 100th of it, 100th of a 60 million base pair genome is still a lot of bases, and probably is perfectly adequate. So we will have a publication soon that says how much of the genome suffices to resolve these questions in Europe. All it takes is some samples from India to run a similar study to see how much do you really need. No one should do more, and no one should spend more than the problem actually requires.
okay, thanks, man. So I think the very first step would be to generate some baseline data to further work on it, means, and see how much variation we do have. And we are assuming or expecting, based on the previous literature, that there will be more as compared to, but yep, I agree with you that it needs to be answered and see how variant isolates we do have in India. Okay, so there is another question for you, Dr. Benjamin.
Yeah, do only boar or pork spread this or other meats too, as India's nation depends mainly on poultry, goat meat, even if, pork, we forage cereal feeds, and what are the chances of infestation this organism spreads from animal to animal? This is an excellent question. Um, so I would say the poultry and the goats should not have trichinella because they should not be eating meat. I'm saying should instead of don't because there are European societies that are fond of eating horse meat. And horses are supposed to be vegetarians. They're grazing animals. There should be no trichinella there. And yet, in France and I think Belgium, there have been some outbreaks of human trichinellosis attributable to consumption of horse meat. And what seems to be going on is that old horses in Eastern Europe that have outlived their principal economic purpose have been fed meat in order to increase their weight, improving their sale. And it turns out that a horse will eat meat if you give it that protein source. It's not a good hunter, but it can eat. So when I say the chickens and the goats should not be contributing in any way, it's a should rather than a don't, because until you look, you won't know. But then any wild carnivore or omnivore should be suspected as a potential reservoir or vector of transmission. I mean, in Europe, foxes and bears are important hosts for trichinella, kind of outside of the swine system, and sometimes those overlap. So I would think that some survey of Indian wildlife, including small omnivores and carnivores, and I don't know all of what you have. We have things like raccoons and possums, rather small tree-dwelling mammals who contribute to uh, trichinella transmission. And there's probably literature that I'm not familiar with in India documenting this issue, but yeah, poultry won't be a concern for you.
Yeah. So let me add few other points as far as India is concerned. So Dr. Benjamin, so we do, as I told you, that we did have a few outbreaks, like it was happened in northern part of India. So in Uttarakhand, and where I told me is the boar meat was responsible for this, and there have been studies done because in northeast states in India they do eat pork, right? But they did survey recently in 2019-20, if I'm correct, but they didn't find any sample positive for trichinella. Here in Punjab, we did serological studies. We did some ELISAs a few years back, and we found that means some of the animals they have been exposed to it. But recently, we do have a project where we are working on it, and we see not that a big concern, but we do find some positives in pigs. So yeah, so let me try to emphasize if the animals that don't have exposure to like rodents or they are scavenging on meat or leftover stuff, so they do get infection. And but as far as in India, no reports from, however, no work has been done in goats or poultry. Definitely not a good indicators for trichinella. But we recently in again in Uttarakhand, there was a report in leopard, so the wild animal. So they found it positive for trichinella spiralis. So the wild animals, they could act as source for trichinella in India, as well as a wild boar or pigs if they are exposed to the positive rodents or other animals. They do it by cannibalism too. So yeah, but that was a good question by Simran. Okay, and if there are no other question, so please join me in thanking Dr. Benjamin. Thank you very much, Dr. Benjamin. And before that, I would like to again I would like to thank authorities of our university, the organizing team. Uh, Organizer in team includes Dr. R.S. Olak, who is director of our School of Public Health and Zoonosis, patron of this Dr. Grumman, who is Dean College of Veterinary Sciences, and other staff members, Poonam and Tejeshwar, who are always in helping more to doing all that stuff for these webinars series. And I would also like to thank our Vice Chancellor, Dr. Inderjit Singh, for supporting us constantly for organizing these webinars, workshops, and trainings. And at the end, I would also like to thank all the participants, our undergrad students, grad students, faculty members who are also showing up today to join this webinar and actively participating in that. Thank you very much, and I highly encourage you to join again in the coming weeks. Thank you very much. And at the end, again, Dr. Benjamin, thank you very much for sparing your time, for your efforts to making this very wonderful presentation and talking on this topic. Uh, thanks a lot. Thank you very much.
Thank you. Thank you all for attending.
Thanks, Ben. Bye now.
Bye.